Okay, so it's why don't we do like a little? I'm gonna ask you some questions for just like some very brief review, and that way we have some, a little bit of everything in this presentation. Okay, so all right, let's talk about. Do you have any questions about water? What are you seeing in this picture? What's happening in this picture? What are these dots? Hydrogen bonding, right? And why do we see this in water? What's special about water that allows it to do this? Never? It's the shape of the molecule. What do you notice? See, one end is positive, one end is negative. So the hydrogen end is positive, the oxygen end is negative. What property does this give water? Gives it a few properties actually, but give me one. Because water is polar and it can form these hydrogen bonds, I'll give you one. One property is it's cohesive, which means that it what? What does it do? It can stick together. So here's an example of an application for this. A tree. Okay, a tree is really high, and it has, it has leaves at the top. This is my really ugly tree. So, some trees could be several hundred feet high. How does a tree get water to its leaves? How does a tree get water from the ground up into its leaves? What's that? Yeah, but how does it work? It's an interesting question. In fact, it's because water is actually cohesive that it can actually do this. What happens is water actually can evaporate out of the leaves. And when it evaporates out of the leaves, it actually pulls. You got to think of water, because water sticks to water and it's cohesive. You can think of it like a chain. So as you pull water out the leaves, it actually moves up the tree. And that only happens because water is cohesive. And the other property is it's adhesive, so it can stick to the sides of the walls in the tree. Like, you remember what the names of the vessel is that contains the water in a tree? It's called the xylem. So we can think that the water can stick to the sides of the xylem, so it's adhesive. Water is also cohesive, which means that you can pull water like a chain, and it can actually literally go up the height of a tree. Okay, so we talked about water. We talked about how it's very important. We talked about how you make big molecules. And you remember what the basic idea is, right? The basic idea is if you want to make something bigger, you have to take away water. So, good. Okay, we talked about that. We've discussed examples of that. So, for instance, in the making of maltose is what? 2 glucose molecules. You can see the water is highlighted here. Take away water and you get maltose. The name of the bond between 2 carbohydrates is called a glycosidic linkage, okay? We talked about, dehydration reactions. The opposite of a dehydration reaction, if you want to take a molecule and break it, it's called a hydrolysis. So if you want to split maltose, you have to add water, and that happens during something like digestion, right? We talked about isomers. Isomers are same formula, the different arrangement. So for example, here we have, you can see butene has the formula is the same for both of these molecules, but the arrangement is different. We talked about how in thalidomide, That's an example, a tragic example, a reminder of an isomer. You can see here that these two molecules are the same, but we can see right over here that there is a difference in the way that it's arranged. If we compare these two over here, you can see that they've been flipped. And this is an example of an isomer that had caused, one version caused morning sickness and the other one caused birth defects. Okay? So we talked about isomers. Here's an example of a mirror image isomer. And we said living things can only use one of these two versions for sugar, and that would be the D version on the right. For amino acids, it's actually the other version, the left-handed version. Okay. So we talked a little bit about carbohydrates. Then we turned our attention to fats. So if we scroll down to fats. So how do you make a fat? Again, same idea. Take the two components of fat, glycerol and fatty acids, and you remove water. Okay, take away water from them. Now, I don't think I gave you the name of the bond between glycerol and a fatty acid chain. It's called an ester linkage. Okay, so it's called an ester linkage. All right, but the idea is the same. Take away water. So fats, we said they're made up of glycerol and fatty acid chains. So if it's 3 chains, it's a triglyceride, 2, it's a diglyceride, 1, it's a monoglyceride. An unsaturated fat is one that has carbon double bonds. So if it's got many carbon double bonds, it's poly. 2 or more, it's poly. If it only has 1 double bond, it's a monounsaturated fat. Now, a saturated fat has the maximum number of hydrogen atoms, and we talked about how you can see that because it's straight and linear, it's often at room temperature a solid. Okay, whereas the bend in the unsaturated fat causes the molecules to spread out, making it more of a liquid at room temperature. Then we asked a really interesting question about, because there's a correlation between saturated fat and melting point, and then unsaturated fat and melting point. Because the saturated fat is a solid at room temperature, and unsaturated fat is a liquid at room temperature. We talked about plants, right? And plants have a combination of both saturated and unsaturated fats in the cell membrane, which makes the cell membrane not really a solid, not really a liquid, more like a jello, kind of in between. So talk about plants are grown in warmer climates. The problem is the cell membrane becomes what? Too much like water, because the fat becomes too much of a liquid. So the plant can compensate by making more saturated fat, right? Which makes oils from hotter climates typically more unhealthy than oils from cooler climates. So we discussed that. We talked about some other examples of fats that don't look like the glycerol with the fatty acid combination, which is the steroids like cholesterol and cortisol that have this form. And then we left off talking about proteins, okay? So proteins, like this one is amylase. We said they're very complicated molecules, okay? Here are some examples of what they do. So in defense, think of antibodies. For hormones, hormones could be made from fat, like you saw cortisol or estrogen. But hormones could also be made from protein, like insulin. Here's the building block of a protein. It's called an amino acid. Same idea. You want to make a bigger molecule. You want to put amino acids together. You got to take away water. Okay, and you can see water is highlighted over here. Now, the name of the bond between 2 amino acids is called a peptide bond. Okay, so this is an example of many amino acids strung together. So there are many peptide bonds. This is called a peptide. This is now here's the thing, the protein, we talked about the relationship between shape and function. Often there's a correlation. And it's really important to protein. The protein shape is very, very important, okay? So let's talk about shape. So to understand shape of a protein, you have to understand that a protein is organized into like these levels, okay? The first level of protein we said is just the list of amino acids. So like, for example, in this list, we can see over here that the first amino acid is lysine. Lys, followed by valine. So over here, valine, okay? And what determines this list? Where does the information come from to put down this list in this particular order? It comes from your DNA, your genes, right? So there would be a gene that, tells a cell that when it's making this protein, that this is the order of amino acids. Lysine's first, valine's next, okay? And at the end is leucine. You can see at the end over here is leucine, right over here. Okay. Now, that primary level can do a few things. It can coil, and this depends on the amino acids, right? So if you have a particular set of amino acids in this area, they may coil. And we talked about the coiling can give the protein elasticity, because if you look at its shape, kind of reminds me of a slinky. But maybe the region in that chain doesn't coil, maybe it folds up like this, and this can give a protein strength. Okay? Now, you'll notice that the actual polypeptide itself has a shape. So the coiling and folding is called the secondary level. But the actual shape of the protein, that's what's known as the tertiary level. That's the third level, okay? So the overall shape here, so you might have some coiling and folding going on. So maybe this area is coiled, maybe this area is folded, but the overall shape is the three-dimensional level. Sorry, the third level, okay? Tertiary level. And it's held by 4 types of bonds that you're seeing here. So here is a hydrogen bond. Here is a bond between 2 things that do not like water. So it's called a hydrophobic interaction. Okay. Here is a disulfide bridge. And here is an ionic bond. All right. So here, this is the same thing that happens in water, right? It's basically the hydrogen end that's slightly positive is facing an oxygen end that's slightly negative, so there's an attractive force there. Here, the attractive force is, well, this protein is sitting in water, so we know that things that don't like water stick together, so that's that interaction. Here, there's one type of amino acid called cysteine, and it's the only one that has sulfur in it, so this is cysteine over here. I'm just going to label that as C. This is cysteine over here. If they face each other, you can get what's called a disulfide bridge. Okay? And that's actually the strongest bond. So this one is actually the strongest one. Okay? This is the strongest bond of the 4. And then you have a bond where one amino acid is, you can see, has a positive end, and the other one has a negative end, and if they face each other, you get an ionic bond. Now, let me explain what these things are. So you see all this stuff here, this. Those are all the R groups. So if you go back to an amino acid, remember, an amino acid, they're all the same except for what? Except for this part here. This is the R group. Okay? So here, what you're seeing is that, some of the R groups are non-polar. Some of the R groups are positively charged, like this one over here. So this R group on this side would be positively charged. This R group would be negatively charged. Okay? This R group is polar, so is this one. So the idea is, if I have my polypeptide chain, and this amino acid over here faces this amino acid over here, and if this one is positive and this one is negative, we know they're going to be attracted to each other. Okay? So, again, what determines that this amino acid is here? And what determines that this amino acid is over here? It's that information from the DNA, right? Now the question is, okay, they're there. If the R groups can form a bond, the bond will hold the protein in its shape. Okay? So this is the third level. Now, what may happen is sometimes a protein only has one chain or one polypeptide, and that's the end of the story. So the one I showed you here, lysozyme, is an example of that. Okay? But a lot of proteins actually are made up of more than one chain. So like, for example, collagen and hemoglobin are made up of more than one chain. So if that's the case, we have what's called a quaternary or fourth level. Okay? So each chain has its own shape, but when we put them together, there's an overall shape to the protein, and that's the fourth level. All right? So you can see collagen has how many chains in it? How many chains do you see in collagen? Yeah, there's 3. Okay, we can see three chains here. So there's here. This is one over here. This is another one over here, and then we have a third one right here, right? Whereas hemoglobin, you can clearly see there's 4 chains. Okay, so this is the fourth level, quaternary level. Now, collagen. Why do we need collagen? It's actually the most abundant protein in your body. I'm pretty sure it's the most abundant protein in your body. And it's used to hold things together. So like it holds blood vessels together, for instance, is part of that. Vitamin C is important in making collagen. Okay? So there's an enzyme that makes, well, there's a bunch of enzymes that work together to make collagen, to make that protein. And they need vitamin C to do it. Okay? So what's a disease when you don't have enough vitamin C? What disease can you get? Yeah, it's called scurvy. So what happens in scurvy is a person could bleed to death over time because, well, if you need vitamin C to make collagen, you don't have enough vitamin C, you can't make collagen. Without the collagen, if the collagen is a protein that holds things together, well, what happened? Things will start breaking apart, right? So you could get like internal bleeding, which would be really, really bad. All right. Okay, we talked about protein shape, and I'm going to go back to function and form. So let me show you an example of a mutation that changes the shape of a protein, and therefore it changes its function, or changes its behavior. So, red blood cells. What's the normal picture for normal red blood cell on this slide here? What's normal? The one on the left, right? Normal hemoglobin. What's wrong with the one on the right? What's wrong with the shape? Not round. Why do red blood cells need to be round? Very easy, so they can roll through your bloodstream. Otherwise, they get stuck. Someone who has sickle cell anemia, blood flow is compromised. Not good. Okay? We need your blood to flow normally. That's why your red blood cells are round, so they can roll through your blood vessels. Now, in sickle cell anemia, it comes down to one little mutation, And you can see it's highlighted here. The mutation is this amino acid over here is substituted for this one over here. Okay? And the thing is, sometimes you get a mutation, there's no effect. But in this case, there is an effect because the type of amino acid that you're changing has a different R group. Glutamic acid has an R group that is polar. Valine has an R group that is nonpolar. Now that's significant because if you go back to your image, and I have a lot of stuff on this, so let's erase this. That's kind of messy. Okay. But let me explain maybe with an easier example to understand. Why is it that a mutation, in this case, produces an effect, okay? So I'm not going to talk about, well, actually it doesn't matter. Okay. So like, okay, here. Why is there a bond here? What's going on here? Why is there a bond here? Right, because these two molecules are polar. This amino acid over here has an R group, and the hydrogen in the R group is slightly positively charged. This amino acid over here has an R group, and the terminal oxygen, the oxygen at the end, is slightly negatively charged. So they're attracted to each other. But what happens if we change one of those amino acids and we made it, instead of polar, we made it maybe nonpolar? What would happen to this bond? Would it be there? It wouldn't be there. Maybe a more dramatic example. Now, so that's what happens in sickle cell anemia. A more dramatic example is, what if a mutation, so here's the amino acid over here, this is the R group, okay? Imagine we change this amino acid so that the R group is negatively charged. What would happen to this bond? Not only would it not be there, there would be repulsion between those two amino acids, right? So what happens in some cases, so in sickle cell anemia, if you change the right amino acid, what could happen is it could disrupt the bond that holds that polypeptide in place. And if that bond gets disrupted, what do you think will happen to the shape of the protein? You think it would be the same? Probably not. So in sickle cell anemia, this is exactly what's going on. Okay? That little, it's a small change, just one little mistake. But because it affects a bond, and that bond is critical to the shape of the protein, it changes the shape of the protein. Now, can you think of a scenario where a mutation could change an amino acid and have no effect? So what do you think would happen if we changed this amino acid over here to a different one, but the R group is still negatively charged? What do you think? It probably will have no effect because it's still the same type of amino acid. It's not the same amino acid, but it's the same type. For me, an analogy would be like if I said the word cat and I spelled it with a C or a K, it's different. But it sounds the same. So it might have no effect in the way you interpret the word cat. Okay? But if I spelled the cat with an R, it's not cat anymore, it's rat. It's different. That might have an impact. That would be like sickle cell anemia. Okay? So, sometimes a mutation, if it affects the bond, could have an impact. Sometimes the mutation will give you the same type of amino acid and have no impact at all. We'll explore that in more detail. Okay, when we there's a whole section on mutations in this course. Okay. Domains. So protein is pretty complicated. All right. we can look at a protein and we can zoom in on it, and a protein may have what are called domains, okay? And domains are areas in a protein that have specific roles. So here's a protein called STAT, okay? And this protein is color-coded. You can see I'm seeing some green up here. Here's some green. Here's some blue. Here's some red. Was that yellow? Was this purple? These different regions, these domains, have different functions, okay? So this red area has a role. What do you think its role is? It's to bind to what? Right here. Bind to DNA. So we know that this protein attaches itself to DNA because it has a DNA binding domain. Part of that protein has a region that attaches itself to DNA. Okay? Now, we know that this is a type of protein that sticks to other proteins. And how do we know that? Because if you look at the proteins, this little region in green here, that region in green is a protein binding domain. So we know that it also interacts not only with DNA, but it also interacts with proteins, with other proteins. We'll explore this in more detail. We'll talk about domains a little bit later on. But I just want you to understand that proteins are pretty complicated, and we can actually look at the protein itself and divide it into think of like geography, like different areas, and those different areas could have different roles. Okay. Domains are really important to a protein. Sometimes they're conserved universally. Now, what that means is that you can look at a domain in a protein from any organism on this planet, and that domain is virtually identical. So this, if my memory serves me correct, is the domain from a protein that builds the heart. I could be wrong, but I'm pretty sure I'm right. And the protein has a unofficial name called Tin Man. Now I want you to think, why would a protein that helps build the heart be called Tin Man? Because if you think of the Wizard of Oz, what did the Tin Man want? He wanted a heart. So this is just a little stretch. The letters represent amino acids. So L is one amino acid. Q is a different one. If you remember before, I showed you a list of amino acids, but the list was made up of 3 letters, right? So like GLY is glycine. But we can abbreviate an amino acid into one letter. It's just easier to read. You don't have to know what the letters stand for. Don't worry about that. But each letter, the basic idea is each letter stands for an amino acid. And more often than not, each letter stands for only one amino acid. Okay? So what you're seeing here is a list from a fruit fly, a mouse, and a human. And if you line up the list, you'll notice something very interesting. They're almost what? Identical. In fact, the second one is the mouse, the third one is the human, and you can see they are identical. And if you compare a fruit fly to a mouse and a human, they're almost identical. You can see there's only a few little differences. They're underlined here. So if a domain is conserved and it's identical, whether it's a human, a mouse, or a fruit fly, it tells you something really important. In biology, it's a simple truth. If something is conserved, that means it can't afford to be changed. Cannot change. All right? Its role is so important that any little change in that role would probably kill the organism or have a really negative impact on the organism. The analogy I often give my students is, I want you to think of a key. Okay? If you think of a key, imagine the key is like a protein. So here we have a key, right? And we have the teeth of the key. Okay, here. And a conserved domain is like the teeth of a key. What happens if you change the teeth of a key? And we were trying to put it into a lock. So here's my lock. Now this key works, but imagine we change the teeth. Okay, we maybe change it. Ooh, change it a little bit here. Change a lot here now. Maybe we change it so it looks different. Will this key work anymore? Probably not. So if a protein has a domain, and the domain is pretty much the same throughout all these organisms, it's telling you that that domain is so important that it can't change. Because it behaves like the teeth of a key. But we'll look at mutations later on in this course, and you see that there are other areas that can change a lot. So those domains, or not those, not shouldn't maybe say domains, but those areas of the protein, maybe not so important. Maybe they can change, and it will have no impact. What part of a key could you change? And it will have really no impact on the way the key works. Is that? Yeah, this part over here, right? If we change the handle, maybe we, make it different, or change the color of the key. There are certain things we do to the key that have no impact. Okay? And we'll look at mutations more in detail. We'll talk about these areas that are conserved. Okay? In more detail later on. Prions. You ever heard of prions? No? Okay. So a protein shape is very important. Change the shape of the protein could change its function. So prions are proteins that do just that. They have a particular shape. So here's a representation of a prion. And what they do is they interact with a normal protein. So here's my normal protein on top here. Okay? It interacts with that protein, and you can see what it does to it. Changes its shape. Problem with this is if that protein is an important one, maybe it's a protein that helps the nervous system work. If I start recruiting that protein and changing its shape, and it becomes abnormal, and it stops working, that could have a really bad impact on my nervous system. So how many of you have heard of mad cow disease? Okay, this is what's going on in that. In mad cow disease, prions are recruiting normal proteins, altering their shape. So what happens is all these proteins that are supposed to help the nervous system work fail over time. It's called mad cow disease because when it was observed in cows, they thought the cows were crazy because of the way they were behaving. Not that they're crazy, it's just that if their nervous system is failing, you'll see symptoms. And an individual can't coordinate themselves properly because the nervous system isn't working. Okay, so here's another clear example of change of protein shape. Just like in sickle cell anemia, change its function, right? Change its behavior. All right, so that was proteins. We're going to talk more about proteins because we're going to do a whole unit on proteins, and when we look at genetics. Okay, so here's a protein. Again, and you can see in this protein, this is an example of the quaternary level. The different colors tells you that it's made up of different chains. So this is the fourth level. We could look at one of the chains, and that would be the third level, because each chain has its own shape. And if you look at the chain, you can see the secondary level too, right? What do you see over here? What's this doing? It's coiling, right? So often the coiling is represented like this, which makes sense. This, you see these arrows? I don't know if you can see these arrows. These arrows represent the folding, that beta pleated formation. And where you see lines, that's nothing. There's no coiling, no folding. All right? This protein has a particular function. Guess what it makes? guess what this protein makes? It's called DNA polymerase. Guess what it makes? It makes DNA, which is our next molecule. Okay? The last molecule, we'll look at our nucleic acids. So nucleic acids are things like DNA or RNA. We know DNA contains what are called genes. And the way it works in genetics is the DNA gets converted into RNA. So the DNA over here gets converted into RNA, which then gets converted into a protein. So there's a connection between DNA and protein. The information to make a protein comes from the DNA. Okay? And we talked about that already. So here is the basic building block of a nucleic acid. This is called a nucleotide. It's made up of 3 parts. It's made up of sugar, a carbohydrate. It's made up of a phosphate group and what's called a nitrogenous base. Okay? This is DNA. It's double-stranded. We'll explore and we'll talk more about DNA later on. Again, I'm going to spend a whole unit on DNA and protein and genetics. Okay? You can see the 3 parts of a nucleotide. So here is my sugar. Here's the base. Here's the phosphate group. Okay? You can see hydrogen bonding here between the 2 strands of DNA. So here's the thing. Hydrogen bonding is really important. We see that it's important for water. It allows water to stick to water, which means trees can pull it to the top of the leaves. We saw hydrogen bonding in some other macromolecules like proteins. We see it here too in nucleic acids. Very important type of bond. Okay? So here's DNA. We know it's double-stranded. We know there's what's called complementary base pairing, which means C and G pair up and A and T pair up. Okay? Well, again, we'll talk more about that later. Here's an example of a mutation. Okay, this is a neat example of mutation. That hideous dog, the black one there, the difference between the major difference between these 2 dogs is a mutation in a gene that basically tells the body to stop making muscle. Because what happens is there's a point where your body says, okay, you have enough muscle, right? So there's this protein that says stop making muscle. What happens if there's a mutation in that protein and it fails to tell the body or fails to tell the organs to stop making muscle? Well, guess what you get? You get a heck of a lot more muscle. And so this dog over here comes down to a mutation in a gene that encodes this protein, and this protein fails to prevent the building of muscle. So you get much more muscle. So the reason why I'm showing you this is because we're going to talk about the interaction between DNA and protein. Okay, because remember, the information to make a protein comes from the DNA. And we'll talk more about this, okay? So here's RNA. RNA is very similar to DNA. There's a few differences. So if we go back to this picture, one difference is in the actual sugar. In RNA, we have OH right over here, whereas in DNA, we have H. That's why DNA is called deoxy. Right? That's why you have the deoxy part in DNA, because it's missing one oxygen. Okay? So here's RNA. Okay? So RNA ribonucleic acid versus deoxyribonucleic acid has one extra oxygen. You can see it. The other difference is instead of the letter T, you'll notice there is no T here. It's U, so U replaces T. And a third difference is typically this is single-stranded. whereas DNA is double-stranded. Okay? So here are some of the differences. Extra O, there is no thymine, it's single-stranded. And the fourth thing is the RNA just usually has the genes, and we'll explore that in more detail when we get to genetics, okay? But I don't want you thinking that nucleic acids is only about genetics, because nucleic acids can be used for making energy. So here's an example of ATP. This is the molecule that allows you to store energy. But if you look at it, what does it look like? What do you see here? What's this? There's your carbohydrate. There's your ribose. There's your nitrogen base. And there's your phosphate. It's three of them. And if you think about what does it look like? Well, it looks like this. Right? It looks exactly like that. So nucleic acids are used to store energy, but they're also used to make energy. Sorry, not store energy. I meant to say nucleic acids are used to store information, but they're also used to store energy. Okay? They're also used in communication. So this is a molecule that's used in communication between cells. Okay? But again, if you look at it, what do you see? You see the three parts, right? You have your nitrogen base, you have your ribose, and you have your phosphate. So this nucleotide is not being used to build DNA or RNA for storing information. It's used for communication. So here's an example of that. So this is a picture of a bunch of unicellular organisms that will come together. It's a type of fungus, right? That will come together. I think it's a fungus. I'm trying to remember actually if it's a fungus or not. But the idea is that these molecules these cells will come together when the environment is very harsh, and they will form a structure. And this structure comes together when this chemical over here is released. So this is the chemical that is used for communicating. Okay, and you'll see this chemical when we get to the section on hormones. All right, and this is the last one. This is also used in making energy. But again, what do you see here? What's this? Carbohydrate, nitrogen base, phosphate. This is made up of two of these, so it's like a dinucleotide. Okay, you can see there's two of them. But the idea is nucleic acids are used for three things, basically. The big one that we're going to look at is storing energy, but it's also used for making it. Sorry, I meant to say, I wanted to do it in order. They are used for storing energy, but the order I wanted to say it in, we typically think of them as storing information, but they're also used in storing and generating energy and in communication. Okay? And I'll leave you with one last thought. Going back to this picture. Okay, last thought. And then we're done. Woohoo! What was this again? This is DNA polymerase. What is it? Is it a carbohydrate? Is it a fat? Is it a protein? Is it DNA? What is it? What is it? If I put this picture on a test, they said, what is this? Based on its shape. And I didn't even give you a name. You would tell me it's a what? It's a protein, because it's so complicated, right? And I asked you, where does the information come from to make this protein? And you told me it was what? There's your DNA, right? Okay. Now, what's interesting about this protein is this protein actually makes DNA. That's what this protein does. So, we can think of a problem here. Because if this protein makes DNA, but we need DNA to make the protein, I can ask you a really simple question. Which came first? And that's where we'll end, and we'll talk about this next time. So that, my friends, is... Oh my god. Did this not tape? Oh, it did tape. Ta-da! The end.